Okay, talking about this technology, what is actually organ on a chip? It is actually, you call it a small chip that have all the organ in it, where you can test new drugs on this organ without using animal and without using human. So it's called in vitro because we use cells. We take like stem cells, propagate, grow it into organ. So you have like it could be heart or could be other organs, skin. So normally we test on animals, but then there were some reports saying that even though it's successful in animal testing, it sometimes fails in clinical trial on human. So now we resort to something before we go to human testing on the population. We use this kit which is representing human because it is using human cells into organ. So we test the new drugs and if it's already shows some effect or good effect, then we can proceed to the clinical trial. But then if it has adverse effect, we will go to back to the design of the drug. So it's in the middle before we go for clinical trial. So in line with the direction now, like the EPAA for animal rights, alternative to animal testing, and also FDA direction, they want it to eliminate animal testing. So by 2035, there will no animal testing anymore. Because one of the components that we want to combine with organ on a chip is the analytical part. So if you have the system model working, but then how you actually analyze it? So you need to have some components that actually work together with the organ on the chip to actually extract the responses from this organ towards drugs. So we call it bioinformatics. So there are data that be analyzed and make a conclusion, decision making. So this is important. I mean, normally if you're talking about testing, you need to have a decision making. Whether it works, it's toxic, it's not work, it have adverse toxicity. So we have organizations like OECD harmonizing the method using cells. The easiest thing to set up a guideline be on the toxicity, because this kind of test will be eliminated in 2035, right? So we want to be at least doing that. We see on, like I say, liver tox, all those things. At least we try to champion on that, even though it's for organ on chip only, not human on chip yet. So maybe at the first stage would be that, like hepatox. Okay, liver tox is a common problem, right? If you take drugs, what are the organs that actually do the digestion of drugs? It would be liver. So that is the main problem there. Even on cancer situation also, it's liver, one of the top. So sometimes you consume a lot of traditional medicine. So normally the doctor say it's not good because it will affect the liver. So meaning the most affected organ would be the liver. So maybe the first template will be the liver toxicity template, hepatox. And then from there we can develop, because it needs a lot of round robins. Round robins is like validation stage, whether this template works for different drugs, and it should be tested by different organizations, not just us. SIRIM become the first, the core, and then we might have different testing punya facility. So they will be testing the same template, but different people testing it. So once you got the consensus that, you can go for standard development, harmonization, and you can actually send this to the OECD for them to evaluate. So that the stage, we have the local first, consensus, international consensus, and then they will compare with other template, and there will be harmonization, and you'll be part of it. How to develop a clear regulatory pathway. That is very important, because where we want to go after this, right? So we want to see how the regulatory bodies see this. Is there any potential or not? Because they will guide actually the translation of this research into clinical application, into market, into the public, right? So the point is regulatory. Yeah, that's the main point.